Primary immunodeficiency of T cells. People with no T cells or poor T cells function are susceptible to opportunistic infections. So what is opportunistic infection? An opportunistic infection is an infection caused by pathogens, for example bacteria, virus or fungi, that take advantage of opportunity that not normally available, such as a host with weakened immune system. So what are the few example? For example, patient with HIV open opportunistic infection like pneumonia and tuberculosis. Because B cell function in humans is largely T cell dependent, T cell deficiency also result in humoral immunodeficiency. That is, it lead to a combined deficiency of both humoral and cell mediated immunodeficiency. Some of the diseases that caused by primary T cell deficiency are severe combined immunodeficiency or SCID, adenosine deaminase deficiency, purine nucleoside phosphorylase deficiency, MHC class 2 deficiency, Wiskott-Aldrich syndrome. These are some of the most common diseases caused by T cell deficiencies. So now let's talk about SCID or severe combined immunodeficiency. Immunodeficiencies that affect both humoral and cell-mediated immunity are called severe combined immunodeficiencies or SCIDs. SCID result from impaired T lymphocyte development with or without defects in B cell maturation. So these SCIDs can develop due to the impaired function of T lymphocyte. So in that case, B cell might work or might not work. Okay. So with or without a defect of B cell maturation, impaired T cell or T lymphocyte development can lead to SCID. SCID is associated with defected cytokine signaling. So cytokine signaling is very important for our bodily function. Cytokine signaling is an important part of body regulation. Most cytokines are cell-secreted proteins from glial cell in the nervous system and are necessary for intercellular signaling. Most cytokines are local regulators that alert and active lymphocytes. Some cytokine signaling pathways involve hormones such as growth hormones and leptin. So you see, this is a very important pathways for regulating body function. A cytokine signal can lead to dormant lymphocytes. The receptors on the lymphocyte recognize the signal and activate it. Those cells are specialized to recognize certain antigens. The combination of macrophage and activation of lymphocyte through cytokine signaling help keep the body in homeostasis or the proper internal equilibrium. So when defective cytokine signaling initiated in our body, it can lead to SCID, like many other diseases. Premature defect of the T lymphocytes due to accumulation of toxic metabolites caused by deficiency of adenosine, ADA or purine nucleoside phosphorylase. We will see details mechanisms of these two enzymes in next slide. Defective VDG, rearrangement in developing lymphocyte caused by the maturation in the genes for RAG1 and RAG2. So at this point, we are well aware of the function of RAG1 and RAG2 genes, and VDG was the antigen receptor. Disruption in pre-TCR, that is T cell receptor or TCR signaling during the development can also lead to SCIDs. Defective thymus development can also result in deficiencies of T cells, which ultimately can cause severe combined immunodeficiency. Why it is called severe combined immunodeficiency? Because this disease can affect both humoral and cell-mediated immunity in our body. So what are the clinical manifestations of SCID? SCID is life-threatening infection, have prolonged diarrhea due to rotavirus or bacterial infection of the gastrointestinal tract, chronic respiratory infection. They can also develop pneumonia and meningitis. Infants with SCIDs die of infection by age 2 years unless immunity is reconstituted by treatment. So now let's talk about the role of adenosine deaminase and purine nucleoside phosphorylase deficiency in SCID. So we already know SCID is a rare autosomal disease of the immune system due to genetic deficiency of adenosine deaminase or purine adenosine phosphorylase. Okay, so ADA or PNP deficiency can lead to SCID. So what happened in our body? Deficiency of this purine degrading enzyme. So adenosine deaminase or ADA or purine nucleoside phosphorylase PNP, these are the enzymes responsible for degradation of purine. So deficiency of this purine degrading enzyme, these two enzymes, results in accumulation of DATP or DGTP in T lymphocytes. Deficiencies of this two enzyme, that is deficiency of ADC, can lead to accumulation of DATP, and deficiency of PNP can lead to accumulation of GTP in T lymphocytes. These metabolites, that is DATP and DGTP, inhibits the enzyme ribonucleotide reductase, which is required for DNA synthesis and therefore for cell replication. As a result, T cell die due to toxic effect of high DATP and DGTP. Near complete loss of T cells occur in SCID. Without functioning T cell, B cell cannot also recognize antigen and make antibodies. Thereby, no immune response is possible. So let's summarize what happens in SCID. In SCID, two very important enzymes, that is adenosine deaminase and purine nucleoside phosphorylase deficiency, can lead to increased accumulation of DATP and DTP in T lymphocytes. Due to the increased level or toxic level of these two metabolites, that is DATP and DGTP, they can inhibit an enzyme, that is ribonucleotide reductase, in our body, and this enzyme is responsible for DNA synthesis as well as cell division. So as these two metabolites inhibit ribonucleotide reductase, they also inhibit cell division. Our body faces complete loss of T cell. So near completion of T cells can also inhibit the function of B cell. Due to the near completion of T cell, B cells also cannot recognize foreign antigen as well as they cannot also make any antibodies. Thereby, no immune response initiated in our body. So now let's talk about another T cell immunodeficiency disease. This is Wiskott-Aldrich syndrome or WAS. Wiskott-Aldrich syndrome is an X-linked immunodeficiency disease. Affected males with WAS have small and profound abnormal platelets, which are also few in number, and this leads to a state called thrombocytopenia. WAS can also develop severe eczema as well as pyogenic and opportunistic infection. They have increased amount of serum immunoglobulin E and immunoglobulin A, normal levels of immunoglobulin G, and decreased amount of immunoglobulin A. So patients with WS have increased level of immunoglobulin A and E, normal level of immunoglobulin G, but decreased level of immunoglobulin M. Patients with WS also have T cells with defective functions. They have recurrent infections, usually affecting ear, sinus, and lungs.